modifying quantitative sensory testing for intellectual and developmental disability populations. We titled this Promises and Pitfalls. For those of you who are Gatlinburg regulars, you know some of this work, and we thought we would bundle it to review our progress to date. And those who are new to Gatlinburg, welcome. And if you're new, this beard isn't always this way, but it's been a COVID year. So here we go. What we thought we would do is I would do a short overview of some of the method, this modified quantitative sensory testing 101, so that we don't need to repeat across each talk. And it is a bit of an homage to Dr. Bodfish, our discussant, because this idea came a long time ago when he and I were young and thinking about things like sensory function and severe disability. So we have a set of talks, one with from Alyssa Merbler at U of M, where she's going to give us a data set on Rett syndrome and the application of modified QST, quantitative sensory testing. Chantel, Barney, and myself on an application in cerebral palsy, pre and post surgery. Griffin, Rooker, formerly at KKI, now at Mount St. Mary's University, on work that we've been doing collaboratively in their self-injury neurobehavioral unit and a shared project there. And Breanne Byiers with the work she's been leading in healthy controls as we try to think about referent values and control comparisons. And then finally, as I mentioned, we have Jim Bodfish serving as a discussant. So here's the overview. Just a quick background on sensory testing or quantitative sensory testing. We know there's a series of events from the periphery to central that of a transduction of a signal to get to central and be detected. And it involves basically a functional, intact central and peripheral nervous system to move that signal up. And there's a way to interrogate that non-invasively. Imaging is one way. Some of the other work we do in biopsy is another way. Non-invasively, you can use this technique called quantitative sensory testing that involves a number of calibrated stimuli that map onto different sensory modalities that can be applied in a controlled way. And what this is doing is mapping on to the peripheral neurology and the innervation. As you recall from any intro neuroscience or biology, we have a number of different nerve or axon types that relate to whether they have myelin wrapped around them or not, and then diameter. And that in part determines speed. And for us, in part, our group is very interested in what we call the A deltas and the C fibers, because those are very important in the transmission of pain and temperature. So you can do bedside QST. If you go into an old-fashioned country neurologist, they might have you turn your head and use a Q-tip or a paper clip to poke different parts of you in relation to where you're experiencing pain, often chronic pain conditions or ice, etc. There are more technical approaches using machinery. And for us, we've taken some of the tools that neurology uses at the bedside, and then we've tried to modify. And the essential logic of this is you're applying a stimulus that in known force, in this case it's heat, and you have a person that reports that they can feel it, and then at some point they say it's painful. And so yes, it started warming up, and then a visual analog scale, the person is pushing a button to say stop, that hurts. The issue, of course, that we're facing is what do you do in the context with an individual, a participant for whom language is compromised? So the ability to self-report isn't there. And that's the modification of MQST. So we know that we're quantifying the stimuli, but we are modifying to take into account language constrictions as well as motor. And there are two broad methods of the tool, quantitative sensory testing: methods of limits, a continuous ramp, or method of levels, a ramp up and down. And there are reaction time issues in the limits approach. So we've modified this tool, quantitative sensory testing, to not rely on language. We have proceeded with mapping on to those known modalities. So we usually include a sham trial. Then there's a light touch using a von Frey filament. Then we use a pinprick, so that's acute. And these are mapping on to different nerves that relate to the relay of that information centrally. Cool touch or cold, pressure, deep pressure. We do a repeated von Frey that's relates to a construct called central sensitization, and the idea that normally non-noxious stimuli may get amplified, or painful stimuli are further amplified, and then finally heat. And so we go through a progression. We videotape the individual, both face and whole body in general, and the coding is time locked to the application and the duration of the stimulus. So that's the general paradigm for the approach for all of the papers that are going to follow, so people don't have to repeat that. So thank you very much, and I'll stop there and hand it off to our next speaker. Hi, I'm going to take the first talk. This is a collaborative work that involves Chantel, Barney, who took the lead and she's at Gillette Children's, with Alyssa Merbler, who we're going to hear later talk about her work in RET, but she's also been integral part of this work. Don Simone and David Walk are our local psychophysics and clinical quantitative sensory testing. And this work, which has been funded by NICHD generously for many years, is a long-standing project where we've been following long-term pain as a secondary outcome for interthecal backflow pumps that are implanted for the management of spasticity and improving muscle tone and cerebral palsy. And what we were trying to do is investigate whether we could gain some predictive knowledge about characterizing individuals pre-surgery, before implant, on forecasting how well they would do post-surgery for pain outcomes predicted by that pre-surgical characterization of sensory function. So I'm going to end with this spaghetti and different colored slide, but I'm just showing it right now to get in your head that I'm going to walk through the study and then come back to what I think we learned related to predicting outcome based on characterizing a sensory profile pre-procedure. So the objectives were to establish feasibility that could we do this using this modified quantitative sensory test that I described in that setup part of our symposia, to create sensory profiles that could be coherently subgrouped, and then test whether those profiles related to post-implant pain outcomes. And what's being implanted again is an interthecal backflow pump that has a fair amount of efficacy data for use of improving muscle tone, managing spasticity, and cerebral palsy. So, small sample, just a purposeful clinical enrolled sample of children with CP indicated for this surgery, complex communication needs. Is prospective within subject design before and after ITB implant. ITB is interthecal backflow pump. Pain response was determined by proxy reported pain intensity change. Greater than 50% change in maximum rated intensity was considered to be responsive to the implant for pain management. And then again, we applied MQST to assess behavioral responsivity to those calibrated sensory stimuli I mentioned prior to surgery. Okay, so 7 individuals with pre-surgical pain had MQST differentiated sensory profiles in relation to the ITB pain outcomes and relative to 2 individuals that had no pain. So pre-surgically, and this, I'm going to end with that figure. Pre-surgically, the ITB pain responsive subgroup, so that was 3 individuals whose pain, their maximum rated pain, was reduced by 50% or more after ITB implant. So we have a measure of pain before and after. For those who had a greater than 50% reduction in pain, we're calling them the pain responsive subgroup. And that was 3 of those overall 9. And those 3 showed an increase in behavioral reactivity to an acute nociceptive stimulus, pinprick, and that cold stimulus before surgery. And then there was a pain persistent group, 4 of those 9 individuals that had pain before and pain after, and they had a different sensory profile in terms of reduced behavioral reactivity to cold and repeated von Frey relative to the no pain. So I'm just going to repeat all that in a slightly different way. 9 individuals. There were 2 that had no pain. They became our reference group, and you're going to see that because I put a gray bar around a Z-normalized plot. So that's who we're comparing to. These two that had no pain. And then we said, okay, did anybody have pain going in? And then who had a greater than 50% reduction going out? 3 people did. And then there were 4 group people, pain before, pain after. So that's the characterization of the pain outcome. And then we wanted to look at the MQST profiling of these individuals. So I'm just going to show you that right now. So the top figure shows individual data. Those are individual plots for each connected dot. You know, I want to point. This is weird doing this through Zoom and our whole format this year for Gatlinburg. Nevertheless, I think I can use a pointer. So this is an individual, and that's across those modalities I mentioned in the setup: sham, light touch, pinprick, cool, pressure, repeated Von Fry, poke, 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 poke for 30 seconds, and heat. And then I group, so those are individuals, looks chaotic. And then I grouped them by whether pain persisted, that was 4, pain reduced, we had 3 that had a greater than 50% reduction, so I gave them green, pain's going, red, still red, and then no pain was our gray. And then this is their mean reactivity in each of those modalities, the MQST modalities. And then what I did, and we're being informed by the chronic pain, neuropathic pain research that gets done in this area, is we Z-normalized them based on the 2 that had no pain at any point of what their profiles or what their range of reaction was. Then the language that gets used is whether there's relative gain or loss compared to a normalized comparison. And so I didn't use quote unquote healthy controls. We're going to hear about some healthy control work later from Breanne, but I used disability controls with no pain. So the 2 individuals with CP that had no pain. And this is what I'm talking about when I say the profiling before surgery. So the pre-surgical profile of the 3 people that had a significant reduction in pain post-surgery was characterized by acute by a gain in reaction to an acute nociceptive stimulus, pinprick. And at the same time, the 4 that pain persisted had a response, a loss relative to the no pain controls, where they were less sensitive to cool. So what does this mean? It means that maybe what we don't know going into that surgery is whose pain is going to persist, who's going to have a better pain outcome, who's going to have a worse pain outcome. For a procedure that's relatively common, that isn't for pain, it's for managing spasticity, but that spasticity is chronic and pain is riding along. And right now there's not a good way to predict the pain outcomes for that surgery. So what we're trying to figure out is, could we figure out in advance who's likely to do better than worse in terms of the pain response? And can we use sensory profiling to do that? So this is a very small n, kind of proof of concept, promises and pitfalls. The promises are, could we predict in advance who's going to have a better outcome on pain before this procedure, so that we could think about analgesic management after the procedure? And what I'm saying is, to use their language, the patients that were very reactive to an acute nociceptive stimulus were the most likely to respond. Their pain was the most likely to respond. Those guys who were characterized as being the least reactive, in this case to cool, were the ones where pain was still persisting. So there's some hypothetical mechanisms that could map onto, et cetera, but that's what we're trying to do with this as a tool. There's still a long ways to go, but kind of in the spirit of old Gatlinburg, this was some preliminary data that I wanted to put into this talk or to this symposia to give you a sense of the way we're trying to use this tool. And with that, I'll stop there. Thank you.
So my name is Alyssa Merbler, and as Frank mentioned, I am presenting on some of our work with the MQST in Rett syndrome. So Rett syndrome is a rare genetic syndrome caused by mutations in the MECP2 gene that primarily affects females. Common characteristics of Rett include motor impairment, limited or absent verbal communication, repetitive hand stereotypy, and autonomic dysfunction such as hyperventilation and breath holding. There's evidence that the MECP2 gene and protein is involved in sensory nociceptive processes, with initial preclinical and clinical work suggesting that multiple systems related to pain behavior and nociception are altered in Rett, such as the opioid system, epidermal innervation, and increased levels of beta endorphin. Rett is associated with several painful conditions such as seizures, constipation, and other musculoskeletal problems. However, there's a history of clinical descriptions of delayed, reduced, or even absent responses to pain. However, there's limited systematic investigation to support this narrative. And when surveyed, caregivers reported that their daughters had episodes of pain in the previous week, upwards of 90%, with over 80% reporting that that pain interfered with daily living in at least one area of daily living. Specific to quantitative sensory testing, preclinical Rett models found differential reactivity to tactile stimuli. So there's evidence of hypersensitivity to some, but hyposensitivity to others. So based on this, we wanted to use an objective measure of sensory function to further learn about sensory function in Rett. The overarching goal of this project was to assess repeated biobehavioral measures related to pain in Rett syndrome. This data set specifically is just looking at behavioral reactivity to the MQST. So first we wanted to assess the test-retest of reactivity across 2 clinical visits. And then the second part is identifying associations of that reactivity with factors related to pain experiences using time 1 data. Based on the animal model findings and parent-reported pain episodes, we hypothesized that girls and women with Rett would be responsive to the set of tactile stimuli, and that reactivity would be somewhat stable over time. So 28 girls and women with Rett participated in the larger project, 21 of which had 2 visits, which are used for this data set. The median time between visits was 6.5 months. The MQST stimuli were applied to the tops of hands and tops of feet. For the actual scoring, hand applications were scored together, feet segments were scored together, and then they were averaged for a total behavioral reactivity score for analyses. And this coding system is very similar to what was described in Frank's talk, has been previously used in Rett. So first are the results of the test-retest. So overall, 70% of the variance in reactivity score was systematically accounted for. 29% of that variance was accounted for by the individual. So people who score high tend to score high across all the stimuli and time points. 30% of the variance was accounted for by the stimulus. So reactivity scores differed by that specific stimulus, such as cool scores being lower than repeated scores. We don't see any systematic variation in reactivity between time 1 and time 2, which we would expect to be lower absent because we aren't introducing an intervention. 10% of the variance is accounted for by the person by time. This could be factors such as day of mood differences, health changes between time 1 and time 2, or other unknown factors that affect reactivity. And again, we do expect some variation in the source. We don't expect everybody to react exactly the same across time. Person by stimulus only accounted for 1% of the variance, meaning that all participants had a similar pattern of reactivity across the stimuli. So we really don't see subgroups that have different patterns of responding across the set of stimuli. And we wouldn't expect to see time by stimulus variation, and we don't in the sample. So these are raw scores at time 1. Reactivity is on the Y, and the stimuli are across on the X. Each dot represents an individual. Our overall takeaway is that for this sample, we do see reactivity in response to all of the stimuli. In the actual model, all stimuli except light touch were significantly higher than sham. In our final model, there were interactions for age, clinical severity, number of previous surgeries, and pain interference score from the previous week, so how much their pain interfered across some daily living activities. This suggests that pain experiences differentially influence reactivity. So for example, having more surgeries was associated with higher behavioral reactivity to heat and repeated stimuli, but lower reactivity to the other stimuli. So based on these data, the MQST continues to be feasible in another severe disability population. And overall, we think we're measuring some aspect of sensory function in Rett, helping to further investigate pain and sensory processes using a more objective measure than clinician or parent report. And while test-retest wasn't perfect, we did find that most of the variance was systematically accounting for and in ways that we'd expect. Our current work is being set up to measure and include other related factors such as those potential day of variables like health or mood status, and including smaller and more standardized time intervals between visits. As Frank discussed a little bit, traditional quantitative sensory testing has been able to differentiate those with pain conditions compared to controls, or as in that CP sample, pain versus no pain within the same group. And while that's not exactly what we've done here, we are finding that the MQST reactivity is related to different facets of pain experiences in this sample in red. And finally, using a protocol like the MQST and observational coding systems can help remove some of that bias associated with parent or clinician report, such as the belief of if individuals with Rett have a lower lower sensitivity to pain, as parent beliefs may affect interpretation of their child's behavior during painful experiences and thus influence how they report about their child's pain. However, coding has its challenges, and these are some of the challenges we're still working on. So first is finding the best ways to balance scoring reactivity, ranging from when people have many small behaviors to a few intense behaviors within one coding system, and deciding how do we credit both of those scenarios reflected through the scores and how we do that in a reliable way. And in the same vein, we want to create a system that can be applied broadly. So this includes including a range of potential behaviors, reactivity behaviors that we may see, but also still grounding them in the literature. We also want to be able to have a system that's capable of being used across diagnostic groups, which is kind of what we're talking about here in the symposium. And part of that is considering individual motor abilities. So a small hand movement away from a stimulus in someone with severe motor impairment may be a really large and effortful movement, where someone with less severe motor impairment, that isn't as much work to do. And so how do we balance taking that individual level into account while again still creating codes that we can reliably score? Finally, one of the biggest challenges is movement disorders. Some of our participants that we see have movement disorders like chorea, where we're seeing large arm and leg movements kind of randomly, and it's so far been pretty difficult to figure out, are those in response to the application or are movements more related to that movement disorder kind of pattern. And this is still a challenge that we're working on. So this is just a thank you to colleagues who helped collect and code the data, the Mayday Fund, NICHD, and RettCenter.org for funding parts of the work, and the participants and their families.
Hi, everyone. I'm Griffin. I'm sorry that we're not together in person. However, I do appreciate the opportunity to present some of the work that we've been doing on sensory perception and behavioral function of self-injurious behavior. And you can see our funding right here. So self-injurious behavior is any response that produces or has the potential to produce wounds to the body. Self-injurious SID, as I'll call it, is a major cause of suffering for individuals with intellectual and developmental disabilities and their caregivers. And it varies in commonness across developmental disorders. However, it can be a prominent feature of these disorders. So if you've never seen an individual engage in self-injurious behavior, here's an example of an individual who was admitted to our unit and is engaging in self-injurious behavior. That's the Kennedy Krieger Neurobehavioral Unit. And you can see he's striking himself over the body and the head. Here's some examples of the damage that self-injurious behavior produces. So bruising, you can see these bite marks, you can see the skin picking, producing open sores, and then head banging. So aside from these examples underscoring the dangerous nature of self-injurious behavior, they also make the point that SIB should be painful. That is, it's a reasonable thought, right? Then to think, well, if SIB is painful and people are engaging in it, maybe these individuals don't feel pain the same way you and I do. And so there's evidence for hypo, hyper, and altered sensory perception in work with individuals with intellectual and developmental disabilities, and for individuals who do and do not engage in self-injurious behavior. Unfortunately, I don't have enough time to talk about all those areas, so I'm just going to highlight some of the research to talk about. There are a number of studies that show correlations between self-injurious behavior and ongoing painful medical conditions in individuals with intellectual and developmental disabilities. So in this case, I'm highlighting the Kennedy and Thompson chapter where they describe a patient who had self-interest behavior that was occurring after lunch every day. And they discovered an ongoing medical condition, treated that medical condition, and then the self-interest behavior went away. More relevant to the current study, Symons et al. Conducted a study, and I'm sure others have discussed this method, where an individuals with and without self-interest behavior experienced sensory testing, and evidence was found that individuals who engaged in SIB were more sensitive to tactile stimulation. Moving on to the function of self-interest behavior, using a procedure known as functional analysis, research suggests that the function of SIB for most individuals is social, in that it occurs to gain access to some consequence delivered by another person. Whether that be access to attention, escape from non-preferred events, or access to a favorite toy, the purpose of SIB is to produce a change in the behavior of another individual. And I'm going to refer to this as socially maintained SIB. In contrast to socially maintained SIB, in approximately 25% of cases, SIB appears not to be maintained by a social reinforcer. That is, SIB occurs when the individual is alone. That is, only when the individual is alone, there's nobody around. Or SIB occurs across contexts indiscriminately of what's going on in the environment around the individual. So research that we've been doing for the past 7 years looks at whether there are subtypes within the context of this automatically maintained class of self-interest behavior. And we've delineated 3 subtypes. One is subtype 1, ASIB, that occurs only when the individual is alone. So SIB only happens when there's nobody else around. And that type of SIB is fairly treatment responsive. And the second type of self-interest behavior, subtype 2, ASIB, persists across all assessment contexts. So that behavior happens irrespective of what's going on in the environment, and that behavior is more treatment resistant. Finally, we've identified a third subtype, which is individuals who engage in automatically maintained self-interest behavior, but also engage in self-restraint. So self-restraint is self-limiting behavior that would appear to protect the individual from engaging in self-interest behavior. So for example, if I hit myself in the head, self-restraint behavior might be sitting on my hand so that it's harder for me to actually hit myself in the head. So both those subtypes 2 and 3 are more treatment resistant. Further, research we've conducted looking at wounds produced by individuals with subtype 2 and 3 ASIB suggests they're at the greatest risk for wounds, with those subtype 2 individuals actually producing the most wounds, followed then by subtype 3, and then by social. So given these unique subtypes, it's reasonable to wonder if differences in sensory perception contribute to the difference in the expression of that self-interest behavior across these subtypes. So what we attempted to do was replicate the Symons et al. 2010 study for individuals who did and did not engage in self-interest behavior, and to examine reactivity across those subtypes that I just talked about. Now, these data are preliminary in the sense of drawing meaningful conclusions about these different classes. However, I do think that they show the utility of the modified quantitative sensory testing model. So we had 20 individuals who participated. 5 of those individuals did not engage in self-interest behavior. Those individuals were admitted to the unit for behavior such as aggression. 15 of the individuals did engage in self-interest behavior, and you can see what the distribution of those individuals were. There were no individuals with subtype 1 automatically maintained self-interest behavior. And the reason for that is, as I mentioned, they are more treatment responsive, and we are in a fairly treatment intense environment, so they likely were treated prior to getting to someplace like the neural behavior unit. So the method was we conducted modified quantitative sensory testing, and this was a trial-based procedure that involved a period with no stimulation, then no stimulation again. We measured changes and that stimulation was applied to the left trapezius muscle of the back. And the reason we selected that location was, again, because some of these individuals were aggressive and we didn't want to set the therapist up to be aggressed too. We measured changes in reactivity when exposed to these stimuli that are listed here, and included a sham condition as the control. And you can see some of our stimuli in this slide. So there's a monofilament on the top picture there, and then the pin prick, which did not break the skin in the bottom left there. and then the deep pressure, which was in the bottom right there. So here are the results across individuals who did and did not engage in self-injurious behavior. And it looks like there are some slight differences across these groups. If you look at the cool and the deep pressure, right, it looks like those individuals with self-injurious behavior are more reactive to those stimuli. And these data were actually scored by Frank's group. We ran the procedure, and then Frank's group scored those videos. And then this is the breakdown across those different types of subclasses and subtypes that I've been talking about. So we have our social group, our automatic group with subtype 2 and 3. And what I'm highlighting here is the apparent reactivity of the subtype 2 individual to that deep pressure, where it looks like there really might be something different going on there for that group as opposed to the other individuals. Things look fairly similar for the rest of the stimuli. So there's no evidence that individuals were less sensitive to tactile stimulation, which replicates the study by Symons et al. And to the extent that individuals were more sensitive, it did look like the subtype 2 individuals, those individuals who were most treatment resistant, who produced the most wounds in other studies, actually appeared more sensitive to the deep pressure stimulation. However, that was only a subset of those individuals in that group. So what we're doing now is looking at those 3 individuals who are the most reactive and trying to figure out if there's anything interesting about those cases as well. Future research should continue to look at reasons for difference in sensory perception, both across populations and for particular functional subclasses and subtypes of self-injurious behavior. Thank you.
All right, I'm going to present some data from a typically developing comparison group and a bit of a pivot from the previous talks. So as you've seen, we think that using the MQST protocol can give us some important information about pain and sensory function in IDD populations. However, we have not yet included any typically developing or non-disability comparison groups in any of our studies, in part because it's unclear how well this type of protocol is going to generalize to non-IDD populations. So the goal of this study was to conduct the MQST protocol in a group of typically developing kids and adults to see how they respond to these stimuli, both in terms of self-report of their subjective experience, and then how that subjective experience correlates with the same behavioral measures that we use in our IDD groups. So the data I'm going to show you come from 35 typically developing individuals. They range in age from 5 to 39 years old. There were some additional kids under the age of 5 who also participated, but kids that young generally are unable to provide reliable self-report ratings. So we're going to focus on this age group. As Alyssa mentioned, we're always refining our coding system, and that has delayed coding for this data set so far. So we've focused on the younger kids here for the behavioral measures. Our hypothesis is that behavioral measures will probably correlate with subjective report better in children than in adults. So we wanted to see in this younger group how it would look. So for behavioral measures, we have 13 kids aged 5 to 12. So the same standardized tactile stimuli in the previous studies. The major difference here is that after each stimulus application, the participants were asked to answer a series of questions. And I'm going to focus on just one here, which was asking the participants to rate on a 0 to 10 scale how bothersome they found the stimulus. So for adolescents and adults, they just gave verbal numeric ratings. For younger kids, they were given the option to select a face on a scale similar to the one you see here, and told to pick the face that was closest to how the stimulus made them feel. We chose this language because we didn't want people responding with how painful it was, since the stimuli are not designed to be at a level where they should elicit a lot of pain, but they might be annoying or uncomfortable. So we wanted to get that sort of measure those things that might also be reflected in our behavioral coding scheme. So as with the other studies, we recorded all of the stimulus applications, and those applications were coded by trained raters. Now we're using a modified version of the FLAC scale, which is a fairly widely used pediatric pain scale that we have modified for this purpose. As an exploratory analysis, we also ran all of the videos through Noldus Face Reader software, which is commercially available software that provides positive and negative affective scores for facial affect. So I'm going to jump straight into the results here. This top graph shows self-report ratings across stimuli by age and sex. So females are on the left, males are on the right. Red points are predicted values at age 5, blue are age 10, green are age 20. And so these are our self-report on the top and then behavioral scores on the bottom. Focusing on the top plot, in general, females showed relatively small age-related effects, and pin, repeated, heat, and pressure were the most highly rated stimuli across those, so the most bothersome. Males showed stronger age-related decreases in scores, particularly for pin and pressure, but those same 4 scores were rated as most bothersome for males across age groups as well. The bottom graph shows the average predicted behavior scores for those 13 kids aged 5 to 12 for whom we have coded data. Among the girls, we didn't see any differences in behavioral reactivity across stimuli, but we only had 4 girls in this sample. So it's quite possible that things would have been clearer with a larger group. In the boys, we see patterns at age 5 where reactivity scores for all stimuli except light touch were elevated compared to sham, but at age 10, only pin and repeated were associated with significant increases in behavioral reactivity, suggesting that there may be some age-related effects in terms of how boys express their subjective experiences behaviorally. This plot shows the correlation between self-report ratings and behavior ratings at the stimulus segment level. So each point represents one stimulus segment from one participant. The correlation here is 0.5, suggesting that these features are moderately correlated. When we examined patterns of scores generated from the automated facial affect software, we did not see patterns that were consistent with the self-report ratings. There were no differences between males and females in this analysis, but there were age-related effects. Among kids, we saw elevated scores during pin, cool, and pressure compared to sham, but light touch, repeated, and heat were not significantly different from sham, which is not what we would have expected. In the older group, there were no significant differences across the stimuli. It's also worth noting that many of the segments could not be analyzed by the software because it could not detect a face. This was not an issue with the human coders. So in general, the software seems to underperform relative to the human coders. There were also no correlations between the automated coding scores and self-report scores. Overall, we think these data are promising and that they suggest that the subjective experience of the test is relatively consistent among typically developing individuals, although there may be some age and sex-related effects that could be attributable to biological differences or could be attributable to socializational or cultural issues that are issues in experimental pain research in the general population. Moderate correspondence between self-report ratings and behavioral ratings are promising, but we need larger samples to determine whether differentiated patterns of behavioral reactivity can be observed in those younger girls and in adolescents and adults. It's also unclear whether the factors influencing the relationship between subjective experience and behavioral expression are consistent between typically developing individuals and individuals with intellectual and developmental disabilities. So we're going to have to do some work to figure out how well we can compare across these two groups. So thank you to our funders and all of my colleagues for their support.
Well, that was a wonderful series of talks, and it's a pleasure to have an opportunity to be the discussant, really to see how far this work has come from kind of the humble beginnings when Frank and I first dreamed this idea many years ago. The title of the symposium speaks to promises and pitfalls, and I want to spend most of my time talking about that. But before I do, I want to draw everyone's attention to what I think are 2 really important points. And the first is how difficult this area of research is. And the second is how important, I would say really critical it is for the field of developmental disabilities. So on the first point, how difficult it is, what these researchers are trying to do is a bit like catching lightning in a bottle. They're trying to quantify a qualitative phenomenon. And I thought an analogy might be useful here. It's kind of like defining what math is. We all know what it is and how important it is. So here's Bertrand Russell, who wrote one of the definitive texts on what mathematics is. And he says that math is the subject in which we never know what we're talking about, nor whether what we're saying is true. And then Charles Darwin actually started as a mathematician and found it too difficult and switched to biology, thought that was easier. He said a mathematician's a blind man in a dark room looking for a black cat which isn't there. So why on earth would anyone do something this difficult? Well, that's the second point. This is incredibly clinically significant. No one would argue that really by nature for developmental disabilities, there's this increased risk for pain-related conditions. And we can honestly say that this is an under-researched area. So every grant application under the sun says what they do is under-researched, but this is really under-researched. And it's under-researched, I think, because there's this huge barrier in IDD. And there's this double whammy. That's a technical term. Pain is a subjective phenomenon, and the expression of pain is largely verbal linguistic. So how on earth do you study subjective experiences with linguistic expression in people with cognitive and language disabilities? And this brings me to what I think is probably, to me, one of the most important promises of the talks that we just heard. And that is that what these groups have done is demonstrated, I would say beyond a shadow of a doubt, just the tremendous rigor, a rigorous approach to doing this difficult but critical task. So they're able to define sensory and it's as a proxy for discomfort and pain, to quantify this in a dimensional way and demonstrate some very important psychometric properties, that it's reliable, stable, and even potentially predictive. And I think it's important to note, and we take for granted, but just how much work the basic behavioral observation method is doing here in this approach and how important that is. And that's very apropos of Gatlinburg. But other promises, sort of generally at the 30,000-foot view that I think these papers have clearly shown, is broad applicability of this approach. So we heard about this not only in typical development, but cerebral palsy and in Rett syndrome. And then this movement, not just in syndromes, but to think about symptoms or specific clinically significant behavior and self-injury. Another kind of area of promise, I think, we're all seeing a lot more research on sensory behavior in developmental disabilities, in particular in things like autism. The lion's share, to put it bluntly, of this is done by parent report, and it's not really clear where that's going to be headed. And so this group has demonstrated a much more rigorous approach to do that. I want to mention and sort of, make sure we note, or something that I noted, is that across the tasks, this repeated stimulus condition was the one that seemed to be doing a lot of the heavy lifting, where we saw some very nice effects. And it's interesting to me that of the stimulus conditions in the QST, this is the one that has the sort of central component to it. And so often one of the criticisms of the QST approach is you're just measuring things that are peripheral, and you're not really getting the central contribution. But this repeated stimulus, whether it's sensitization, habituation, learning, something higher order, really is a tremendous addition that these groups have added to this, and it seems to be very important. And then the last sort of big area of promise is that in terms of clinical translation, they've shown that this is an approach that gets to individual differences, the synchronon of developmental disabilities, and that gets to functional subtypes, that is subtypes that mean something clinically, and gets to this sort of promise that we almost never realize, potentially, of personalized medicine. So there's several pitfalls. I'm going to finish talking about each of these, but I did want to sort of bore down a little bit more into some of the promises in each of the specific talks. So in the talk that Frank and Chantel gave in the area of CP, Well, first, across all of these talks, we saw a replication of basic psychometric characteristics of this approach in different populations. So in addition to rigor, we're seeing reproducibility here. And then in the talk on CP, we're seeing the potential for this to extend to treatment research, and the very exciting possibility, if this is replicated, that a pre-treatment QST might predict the kind or degree of treatment outcome. Very important. Kind of related to that, what Alyssa was able to show us, again, extension to Rett syndrome, but a very important psychometric feature, temporal stability. This is a necessary feature of an outcome measure. It has to be temporally stable before it can be an outcome measure. And also something I thought was really promising is the idea that, you know, pain experience isn't equal to pain expression, and you could potentially quantify and begin to study experience, something like the history of surgeries, which she talked about. In Griffin's talk, the extension to SIB subtypes, this very nice integration of a quintessentially behavioral approach, functional analysis, with this, if you will, biobehavioral approach. We're studying a medical physiologic phenomenon in pain through QST, integrating those very nicely. And the take-home message here, really following from much of the work that he and his group have done, very clearly showing us that SIB is not one thing, and that you can use this method to validate these functional phenotypes that could drive treatment decisions. And then finally, in Breanne's paper, applying this, I thought kind of a very neat kind of back translation to typical development that allows you, because you're dealing with typically developing children who can speak and tell you more about their subjective experience, to look at the validity of the relation between subjective and these objective metrics, because in the end, pain is a subjective experience, and this data tells us something very important about that. And so I'll just finish with what sort of I gleaned in terms of some pitfalls in this area, and really this is just areas for future research. The first is, what is sensation in this data, and what's discomfort and pain? And so this is a procedure that's really about sensation. You're delivering sub-pain threshold stimuli. But what's important is that sensation is pain in certain neurological conditions like hyperalgesia or allodynia. And so the question becomes, what is the expression profile that you're seeing in each of these data sets, and how much of this is pain? And so the thought I had is the possibility to add additional contrast groups. One you've done, the typical development group, would be sort of the low end, more to the sensory end of this dimension, if you will. And then potential for neurological conditions where we know that there's pain to act as sort of the pain end. And so other groups would allow us to give that sort of reference to that. The second area is, are we measuring sensation perception, or are we measuring the expression of a sensation? And of course, the coding is really measuring the expressive end of this. But by grading the intensity levels, there's just one stimuli for each of these conditions, and by grading the intensity or the duration of those, this holds the promise to start to press the sensory perceptual end of this and see to what extent expression varies as a function of something like duration. A third area is that the coding approach is summing across a lot of different potential signs of pain or discomfort. And that leaves open the possibility that at the individual level, there may be something very salient, or a small collection of behaviors that are more salient and potentially more sensitive. And that the global coding approach, which you have to start with, may be blurring some signal at the individual level. And so in pain work, this is thought about as individual pain profiles, but this sort of promise of individual differences could potentially be really extended by thinking of individual pain profiles within the data. And then last, as just sort of a bigger issue conceptually here, in these studies, we don't really know what the mechanism is. Sort of the approach here is that there's something that's leading to increased pain sensitivity or increased sensitivity or pain. We don't really know what the mechanism of that is. And until we know that, we're still this blind man in the dark room, to bring back Darwin's quote. And our instinct is to reduce, to think about brain and neurological symptoms. But I just want to close by adding that there's another way to think about this conceptually or developmentally, that pain may be not just a consequence, but also a cause of many of the things we're interested in developmental disabilities. And so there's this approach here, quantitative sensory testing, that could be moved very early in development in prospective developmental studies. And I know Dr. Symons and his group are looking at this too, to see to what extent, now that we can quantify this in a rigorous and reproducible manner, this is leading to important outcomes that we're interested in developmental disabilities. So I'll close there, and again, thank the group for the opportunity to discuss this.